Good afternoon, students. So I welcome you all in the new session of biology. It is very interesting and you are also giving a lot of attention towards these topics. So let us begin with our new topic from the lesson 6 tissues. We already have started with the plant tissues. Plant tissues are of mainly two types. First is the meristematic tissue and second is the permanent tissues. So permanent tissues are also of two types. First is the simple permanent tissue and second is the complex permanent tissues. We have studied detail about the meristematic tissues, then the simple permanent tissues like parenchyma, collenchyma and sclerenchyma, and with that certain protective tissues are also taken care of, right? So we have studied the protective tissues as epidermis and cork. Now we are going to start with the complex permanent tissues. We already have seen first complex permanent tissues, that is nothing but xylem. So today we are going to learn the second permanent tissue and that is nothing but phloem. So what is phloem? It is the transporting tissue which transports the food which are prepared in the leaves of the plant. Okay, so here the leaves, they perform the process of photosynthesis. We already have known about it, that they perform the process of photosynthesis, and as a result of photosynthesis, starch, then water and oxygen are the byproducts. So this starch which is prepared by the plants works as a food of the plants, right? We have studied this. Then oxygen is liberated out through the stomatal opening and water is also given out through the stomatal opening by the process of transpiration, right? We have studied this in detail. Now we are going to see that the food which is prepared by the plant in the form of starch is transported to the various parts of the plants with the help of a vascular bundle. Correct. The phloem is said as vascular bundle. Why? Because it has various cells as vessels. Okay, has various types of cells which are bound together with the help of fibers. So they are called as vascular bundles. So this phloem is the vascular bundle and it transports the food from leaves to other parts of the plants. Materials can move in both directions in it. It's not like only like xylem. Xylem, only upward movement of water is seen, correct? From roots towards the various parts of the plant by the capillary action of water. Yes, when the vacuum is created in the xylem vessels or xylem tracheids, and then the water is sucked up in this xylem tissue. Correct? That we have seen. But here in phloem, what happens? The food is transported in all the directions, not just downward or in upward direction, but in all the directions. Why? Because food needs to be provided to each and every part of the plant. It should be given to the leaves, to the branches, to the fruit, to the flower, to the roots, to the root hair, to each and every minute cell. Food needs to be provided, and that is done with the help of phloem. All phloem cells are living except phloem fibers. So see here, phloem cells are living cells. Like in xylem, only xylem parenchyma was the living and rest all are non-living cells, whereas in phloem all the cells are living cells having the nucleus into it, having the cytoplasm into it, and all the cell organelles, all the cell parts are there in these cells. So they are living cells. Only phloem fibers are the dead cells in the tissue phloem. Okay, I hope you understood this. Now see here, phloem is made up of following 4 types of elements. As I have told you about the xylem, that the parts of xylem are called as elements. Same way the parts of phloem are called as elements of phloem. So it is made up of sieve tubes. The first part is called as sieve tubes. Then the second is companion cell. And the third is phloem parenchyma, and the fourth one is phloem fibers. So these 4 different elements are present in the phloem. What are that? Sieve tubes. Why they are called as sieve tubes, do you know? Because these tubes are porous, having small openings on their walls. So they are called as sieve tubes. It looks like a sieve, correct? Salni in Marathi. We say it as salni. How it has small holes, the same way these tubes are also having small openings on the outer side. So they are called as sieve tubes. So see here, these are tubular cells. Tubular means tube-like with perforated walls. Perforated means having holes on them, small, tiny, minute holes are present on the walls. So they are called as perforated walls. Then they have thin layer of cytoplasm, as these are live cells. So they have cytoplasm. So have cytoplasm into it. Now the second important cell present in the phloem is companion cells. Okay, companionship, the name itself suggests companion means they are living together. So they help in joining the sieve tubes together. So they are called as companion cells. Okay, so these are small elongated cells having thin walls which are not perforated. Okay, so see the difference. Sieve tubes are perforated, that means having thin holes on it. But the companion cells are non-perforated, means they don't have any kind of holes or any kind of openings on them. So they are not perforated. Got it everybody? Sieve tubes and companion cells. Now the third is phloem fibers. As I have told you that these are the only cells which are dead cells. They are just used for the mechanical strength and binding purpose. They actually provide the grip or provide all the cells to remain together. Like how we do when we tie the sticks together. Right? If you will take 3 to 4 sticks and if you tie it with the help of thread, how it looks? The same way these phloem tissues are looking like with the help of these fibers, that they tie all the cells together. So and as they tie it together, it becomes quite tough, quite stiff. That's why it provides the mechanical strength to the tissues. That is the function of phloem fiber, that they provide mechanical strength to the tissue. Then D, that is phloem fibers. So the phloem parenchyma, they help in storage and slow lateral conduction of food. Like in xylem. In xylem also the lateral conduction of food is the lateral conduction of water is done with the help of xylem parenchyma. Same way here in phloem, it helps in storage, as parenchyma are the storage cells. So help in storage and slow lateral conduction of food. Okay, so here phloem provides slow lateral conduction of the food. Lateral means not just up and down, but in right and left directions as well. Lateral means towards right and left directions, that is horizontal directions. So it provides the food, conducts the food to the parts which are present in the horizontal directions of the plant. So that is the function of phloem parenchyma, that they conduct the food to the lateral sides of the plant. Okay, now this process of conduction of food, conduction of these all the material from the leaves towards the various parts of the plant is called as translocation. What it is called? Translocation. That means transporting to the various locations. So it is translocation, the process by which phloem transports the food from the leaves to the various parts of the plants. It is called as translocation. I hope you understood this. That what is translocation? Translocation is nothing but the transfer of food from leaves to the various parts of the plants in all the directions. That is called as translocation. Okay, so phloem performs translocation of the food. For this purpose, energy is needed by the phloem cells. Energy is utilized by the phloem cells in the form of ATP, that we know, and that's why plants need to generate the energy. By the process of respiration during night time, plants generate the energy in the form of ATP molecules, and these ATP molecules are utilized by the plants for the purpose of translocation of the food. And this is how the energy generated by plant is utilized by the plant. So I hope you understood about the phloem, that is nothing but complex permanent tissue. So here we finish with all the types of plant tissues. It may be the meristematic tissues, it may be the permanent tissues. From permanent tissues we have studied simple permanent tissues and complex permanent tissues. In complex permanent tissues we have studied xylem and phloem. Okay, here in this page they have given the diagram of phloem, which is very easy. If you see, they have shown the sieve plates at the uppermost side. These sieve plates are actually for the purpose of entering the food in the phloem tissue. So from sieve plates the food enters in the sieve tubes, and through sieve tubes it is given to the adjacent phloem cells. As it is perforated, from that tiny holes the food is given to the adjacent phloem tissues, and this is how it goes forward in the plant. Then the phloem parenchyma. Parenchyma actually is required for storage of food, where actually the energy is generated, and with the help of that energy the food is translocated. Companion cells are there, as these are very soft and they provide a cushioning effect, so that the food will remain in a particular area for the purpose of distribution. So this is how companion cells are important. And then sieve pores. At the end you can see the sieve pores, and these sieve pores, from there, from one tissue to other tissue, the food is transported through the sieve pores. So this is how the phloem tissue works for translocation of the food. I hope you understood the phloem tissue. Now the next point which we are going to discuss is differences between meristematic and permanent tissues. So see here, meristematic tissues and permanent tissues. It is given in this picture as the differentiating points. So see here, meristematic tissues are cells divide rapidly. That is the function of meristematic tissue, that they are dividing tissues. So here the cells divide rapidly. So the division of cell is done in meristematic tissue, and the differentiation is done by the permanent tissues. So cells are differentiated from the meristematic tissues and normally do not divide. So permanent tissues are differentiating tissues. They differentiate themselves as different parts, and they normally do not divide. Then the second point, cells are undifferentiated and intercellular spaces are absent. So here there is no differentiation when they are there in the meristematic tissues. There is no differentiation. All the cells are one and the same. And intercellular spaces, that means in between the cells, small spaces, small cavities are there. These cavities are absent in the meristematic tissues. Whereas in permanent tissues, cells are fully differentiated. That means each of the cell knows which function that cell has to perform. That is called as differentiation. And they have the intercellular spaces. As they need to go to various organs for forming various organs, they have some cavities into them, which are called as intercellular spaces. So in permanent tissues, intercellular spaces are present. Then see the meristematic tissue, third point, vacuoles are absent. So here, as they don't have to store anything into them, so there is no vacuole in them. So vacuoles are absent, but permanent tissues need to store the food. So the vacuoles are present in permanent tissues. Then see the fourth point, cells have dense cytoplasm. So here the cytoplasm is quite thick, dense cytoplasm. Okay, and in permanent tissues, cells have thin layer of cytoplasm which is around the vacuoles. And then see the fifth point, cells are always living. So here in meristematic cells, as these are dividing cells, they are living cells. Whereas permanent tissues, they may be living or may be dead. Okay, like we have seen xylem parenchyma, phloem, sieve tubes or companion cells, all these cells are living cells. But few cells are dead cells also, like tracheids in xylem or fibers, these are dead cells. So in permanent tissues, the cells may be live or may be dead. But in meristematic tissues, the cells are always living. Okay, then cell wall of its cells are thin. In meristematic tissues, the walls are thin, whereas in permanent tissues, cell walls of the cells can be thick or thin. Depend upon the function of that cell, which function the cell is performing, the walls may be thick or may be thin. So these are the points of differentiation between meristematic tissues and permanent tissues. I hope you understood today's session, where we have studied about the phloem tissue, then the types of its elements, then we have studied the first type, sieve tubes, and second type, companion cells, third is phloem fibers, and fourth is phloem parenchyma and their function. And after that, we have seen the differences between meristematic and permanent tissues. So I hope you understood today's session. Thank you.